इससे स्टार्ट है. Okay, you can see.
Yes, sir. Yes, sir.
Okay. And देखो antibacterial chemicals, bactericidal and bacteriostatic agent. So last class we discussed about the sterility of the different compounds, solid, liquid, ophthalmic and parenteral preparation. But in case of antibacterial agent or especially the antibiotics, if you prepare in the injection for that must be sterile and that will be sterility test done. But what are the antibacterial agents? That means which killed or growth arrest the growth of the microorganisms or especially the bacteria, and which are bactericidal and which are bacteriostatic. Cidal means to kill the microorganism or totally free from microorganisms and it will be killed, and bacteriostatic means the growth or reproduction will be arrested. That is called the bacteriostatic agent. But when it is used, when bactericidal are used and when the bacteriostatic agent used, but what chemical which are used as a bactericidal and the bacteriostatic, and what are the importance and what are the application, where it will be applied and when it will be applied. This is very important. All times the bactericidal is a broad spectrum. So or the in the all the cases you can use the bactericidal. Why use the bacteriostatic? Because there is a some provision, there is a some situation, at that situation bacteriostatic agent and at that time there is a bactericidal agent. Let us we can see what is the agents, and there is a very common terminology you can remember it. It's contain the antibacterial, that means which fight against the bacteria, and the antibacterial agent, those agents or the chemical compound which effective against the bacteria. And what are the classification of the antibacterial agents and bactericidal and the bacteriostatic, and what are the basic mechanism of the bactericidal and bacteriostatic? By this mechanism, bacteriostatic act on this by this mechanism and the bactericidal by this mechanism, and what are the advantage and disadvantage of this bactericidal and bacteriostatic agents. And reproduction of the bacteria, that is called the antibacterial agents. Their reproduction, that means their growth will be arrested and their reproduction will be inhibited. That is called the antibacterial agent, and this agent kill or prevent the bacterial infection by the fighting against the bacteria. Heat and chemical such as chlorine and all the antibiotics drugs have the antibacterial property. Heat is the one of the most important physical parameter by which you can kill the microorganism. But suppose you infected by the some microorganisms, you are heated, microorganism will kill, but you will be also burned. So heat cannot be applied for the any materials. Sometimes there are heat resistance only you can use. Suppose say for glasswares, you can free from microorganisms by the hot air, by hot air. So the heat can be applied or some. So heat, not only the high temperature, much temperature that can be kill the microorganisms, and the chemicals such as chlorine. Say for example, not only the chlorines, other also oxidants, so many compounds which are used as a antibacterial function. Another is the all antibiotics. All antibiotics, that means when are used, I think most of the people, all of the people, you take antibiotics in your life, at this time. So antibiotic drugs having the antibacterial property. Obviously all the antibiotics are having the antibacterial, not only antibacterial but also effective in the others organism also. Antibiotics and the antibacterial both attack bacteria, and antibiotic also known as the antibacterial agent. Antibiotic and antibacterial both attack the bacteria, but antibiotics also known as a antibacterial. That means antibiotic which are acts as a antibacterial agents, but all antibacterial agents are not antibiotics. Am I clear? Antibacterial cannot treat viral infection like the flu and flu. Antibacterial agents, they are effective against the bacteria, not in the fungus, not in the virus. So that's why it is called the antibacterial. But antimicrobial, that means they are effective against any type of any sort of microorganisms. So there is a basic difference between the antibacterial agent, antibacteriostatic agent, bactericidal agent, and the antimicrobial agent, antibiotics. So this basic terminology you can know it, and this question is come in any type of interview or other type of sort of examination. So there is a clear crystal definition of this particular terminology. There are some essential related terms of studying the antibacterial agents are the mentioned here. Follow. Number one, biocides. What is biocide? It is a widespread chemical and physical agent which is inactivate the microorganism. Actually, biocides, that means effective against or fight against or kill against the any viable microorganisms. That is called the biocides. Wide spectrum chemical or any physical agent, not only the chemical or antibiotics, but also the any physical agent, heat, X-ray, gamma ray, which is agent which is inactivate the microorganism, that is called the biocides. And bacteriostatic, that is the property of specific biocides agents which is able to bacterial multiplication, that is called the bacteriostatic. Static means not dynamics, it is a static, that means property of specific biocides, but under the it is also biocides agent which able to bacterial multiplication. Another is the bactericidal. It is also one type of biocides are able to kill the bacteria. Here is the stop the growth of the microorganism or reproduction, but bactericidal means to kill the microorganism. Disinfectant, inanimate object. Disinfectant, what are the difference between the bactericidal and bacteriostatic? But disinfectant is the chemical which are generally used in inanimate objects. Suppose the operation table and the room, aseptic rooms, that can be this inanimate, non-living objects can be sterilized by the disinfectant. If you use the disinfectant in case of the living things, there is a problem, it will be toxic, it is a corrosive action. So that's why disinfectant use only the inanimate object. And the septic, what is the term? Septic, sometimes you can do septicemia or septic. Septic is nothing but a characterized by the presence of the pathogenic microorganism in the tissue. It is called the septic. That means any pathogenic microorganism which is introduced into the tissue and produce some infection, that is called the septic. And this microorganism which is occurred in the blood, that is called the septicemia. It is a very, very dangerous disease and life-threatening disorder. If it is not treated properly, death may be occurs. And number three, the antiseptic. What are the antiseptic? You can use in every daily life, and the borolin, antiseptic, boroplast and others. Antiseptics, especially it is used for the same thing as like a disinfectant, but it is used in the case of living organism, not even inhibit the growth of the microorganism or in the living tissue. That is the difference between the antiseptic and the disinfectant. Disinfectant are the chemical, antiseptic also chemical. Disinfectant use only the inanimate object, that means not living things, but antiseptic which are used in the living things. Say for example, the borolin, boroplast, after shave lotion which are used in the just after shaving, or you cut or burn cut by some things, or a small type of cut in the face or others organ, at that time you can use some antiseptic agents, not use any disinfectant, though the property is the same to kill or the microorganism. But this type of this disinfectant which aggravate the sepsis in your living things, but in case of antiseptic, it can prevent the growth of the microorganism, it can wound the healing. Septic, to keep the free from microorganism. Aseptic condition I discussed, Manasir, I think in case of practical aseptic, if you needed, I discuss elaborately in the aseptic condition or aseptic chamber in case of microbiology. So aseptic, to keep free from microorganisms, that is called the aseptic. This terminology is very, very, I request you to remember it. What are the difference between this the terminology, and this term is very, very important, not only the microbiology, biochemistry, but also the any other pharmaceutical company, they ask the question, because this is a very, very common terms. Be careful, remember it. Antibiotics. This is the most common use of the drugs and most misuse of the drugs nowadays. And naturally occurring or the synthetic compounds which are inhibit or destroy the selective bacteria at low concentration. What are the antibiotics? This is quite different from the antiseptic, from the disinfectant. Especially, it is synthesized from the one microorganism which are effective against another microorganism or inhibit or destroy the selective bacteria at very low concentration, or better, it is a minimum inhibitory concentration. What do you mean by the minimum inhibitory concentration? Suppose one antibiotic, say for example ciprofloxacin antibiotic, which are 200 milligram, which are not effective, 300 not effective, 500 milligram it is effective, 600 milligram effective, 800 milligram effective, and 1000 milligram also effective. Which concentration you used as antibiotics? You can tell me. 200 to 800 different concentration. 200 not effective, 400 not effective, 500 effective, 600 effective, 700 effective, 800, 1100 milligram effective. But which concentration is used?
Sir, 500.
কে বলতে পারবে? Hello? Hello? 500. Why not? You can you use the higher percentage of 500, but below 500 there is no effective. That's why there is no question of use. But in case of higher percentage, above the 500, 600, 700, 800, 900 or 1000, so their therapeutic activity high, but they are not used. Why? Because the toxicity level will be high. So that's why this is the optimum dose or optimum concentrate, but there is no problem. There may be toxic. If ধরো তোমাকে যদি 5 টাকা দিলে কাজ হয়ে যায়, তোমাকে unnecessary 100 টাকা দেবো কেন? 100 টাকা দিলে obviously তুমি অনেক কাজ করবে, কিন্তু এখানে কি হবে? Another problem, there is a produce some toxic. So that's why
কিছু ক্ষেত্রে তো 1000 mg ইউজ করা হয়।
না না, সেটা তার মিনিমাম এফেক্টিভ কনসেন্ট্রেশন 100 mg।
আচ্ছা,
ওকে।
ও
এই নয় যে সব ক্ষেত্রে, মানে প্রত্যেক ড্রাগেরই ডিফারেন্ট রয়েছে, কিন্তু সেটাই ইউজ করা হবে যেটা তার মিনিমাম ইনহিবিটরি করে, মানে যে কনসেন্ট্রেশনের ডোজ দ্য মিনিমাম কনসেন্ট্রেশন হোয়ার দ্য মাইক্রোঅর্গানিজম উইল বি কিল্ড। তার থেকে বেশি কনসেন্ট্রেশন দিলে অবভিয়াসলি ইট উইল বি থেরাপিউটিক ভ্যালু পাওয়া যাবে, বাট দেয়ার উইল বি এ টক্সিক এফেক্ট অর সাইড এফেক্ট উইল বি হাই। সো মিনিমাইজ, আমার তো আননেসেসারি দরকার নেই। দেয়ার ইজ কস্ট এফেক্টিভ অ্যানাদার প্রবলেম, বিকজ তোমার যদি 500 তে কাজ হয়ে যায়, হোয়াই ইউ ইউজ দ্য 100 thousands? ওকে,
ইয়েস স্যার,
this is called the minimum inhibitory concentration. Okay,
ইয়েস স্যার।
যেমন দেখো, in case of metronidazole 400, in case of ciprofloxacin 500, in case of our levofloxacin same group of it is 500 milligrams, and ofloxacin 200 or different types of.
স্যার ওটা 500 এটা 200 কেন?
because they are the concentration, the minimum effective concentration, যে কনসেন্ট্রেশনে এটা কিল করবে সেই কনসেন্ট্রেশনটাকে ডোজ হিসাবে ক্যালকুলেট করা হবে, স্ট্যান্ডার্ড ডোজ হিসাবে বা অপটিমাম ডোজ হিসাবে ইউজ করা হয় থাকে।
ওকে,
হ্যালো,
ইয়েস স্যার,
ঠিক আছে
ইয়েস স্যার।
দেখো প্রত্যেকটা ড্রাগেরই ডোজ কিন্তু প্রায় ডিফারেন্ট, সবারই 500 নয় কিন্তু। আচ্ছা, সে ফর এক্সাম্পল এটা যদিও অ্যান্টিবায়োটিকের কথা, অ্যান্টিবায়োটিকই বলা ডিফারেন্ট ডোজ, যেমন ডোজ যেমন ডিজিজ হবে তার উপরে ডোজ হয়, এবং ডোজটা হবে মিনিমাম কনসেন্ট্রেশন, তার থেকে বেশি হলে টক্সিক হবে। ওকে, অ্যান্টিব্যাকটেরিয়াল এজেন্টস, দেখো হিয়ার ইজ দ্য সাম সোপ। Generally we know the soap is used as an antibacterial agent, and this is, I think this is today you are very much sure the soap is antibiotic. That's why in case of corona, when it start, they all times the news TV and news channels, they shows that they every 30-minute interval there you wash your hands by the soap or by the some hand soaps. So soap is one of the most important antibacterial agent because it acts just like a surface active agent to remove the microorganisms, not killed. Sometimes it will be killed, but another hand lotion then also our sanitizer, you can use this to remove them, to easily remove the micro or kill the microorganism by the hand lotion or by the sanitizers. But another number three is the disinfectant. But disinfectant you cannot use by the hands. Generally, the phenol derivatives and lysol derivatives, these are not used by the living things. These are not normal. These are basically destroy the non-living or inanimate object. Another is the surface spray. Sometimes the operation table or other things or the working area, aseptic chamber, that will be mic on আছে, mute your mic. The surface spray, there is some spraying reagent or some spraying disinfectant or sanitizer. You can use the surface of the surface of the table or operation theater or an others working area. Another is the mouthwash. Mouthwash is also used as a germicidal and the antimicrobial activity. Sometimes mouths produce some breath smell, bad smell. To remove this bad smell, generally the mouthwash is used chlorhexidine or different types of chemical compound and available in the market. So many branded mouthwash, Garland, which is to freshness and breathe and the mouthwash. And another the toothpaste to clean the teeth as well as the kill the microorganisms. So toothpaste is another surface agent which are in the form of paste, or mouthwash is the form of liquid or spray, and toothpaste is used in the form of paste which are cleansing and the teeth or kill the microorganism. Another last one is the antibiotics. This is the most important chemical compound or the it is coming from. They are very important. You write down this, what is antibiotic. The antibiotic are the substances which is coming from or which is collected from the one organism which is killed the another organisms, which is these chemical compound which is collected or source is the one organisms and which are effective against the another microorganism. This is quite different from the others antibacterial agents. Classification of antibiotics, antibacterial agents can classified into five groups of action on the based on the type of action. So antibacterial agent can be classified based on the different class, generally five class, based on the type of action. Which type of action? It is a bactericidal or bacteriostatic. Number two, based on the source. Which source? Where it will be collected? It is an animal source, plant source, synthetic, or mineral source. So based on the source, they are really classified. Another is the spectrum of activity. Obviously, the spectrum of activity, there is a broad spectrum or narrow spectrum. Some bacterial agents which are bacteriostatic but wide range of activity, but some are the less range of activity. Another is the based on the chemical structure, different compound having their different chemical structure. Based on this property, there are phenol derivative, lysol different derivatives, antibiotics different derivatives, another surface active agent. So based on the chemical structure, we are classified on the different antibacterial agent. Finally, and the last is the function. Based on the function, it will be classified into a different class. Those are so based on the function, their activity. First, the type of action. Number one is the bactericidal, and number two is the bacteriostatic. What is bactericidal? Bactericidal means to kill the microorganism, totally remove the microorganisms, and bacteriostatic only arrest the growth and reduce reduce the reproduction. Bactericidal, first, those antibiotics which are actually the antibiotics, those are actually killed bacteria by any mechanisms depending on the antibiotics and inhibit the cell wall synthesis. Especially the antibiotic, based on the mechanism, there are different types of action: cell wall synthesis, protein synthesis, and others we discuss in the next slides. Action, it is irreversible and do not work in the immune system of the host. This antibiotic only effective against the microorganism, not affect the host cell. That means you take the antibiotics, only the antibiotic kill the microorganism, not destroy your host cell. Minimal bacterial concentration, or that is called the MIC, minimum inhibitory concentration, it is required to kill 99.99% of bacterial population. Bacterial population, for example, bacterial used for example, the beta-lactam antibiotics in this class, different types of the penicillin group of antibiotics, penicillin, amoxicillin, all are the beta-lactam. Another beta-lactam group of antibiotics, cephalosporin, cephalosporin, cephalexin, ceftriaxone, and so many cephalosporin, all are under the class of beta-lactam. Beta-lactam is a beta-lactam ring, basically based on the chemical structure, but their pharmacology action are different. That's why there are two class. One is the cephalosporin, another is the penicillin. And under this cephalosporin, so many drugs, 1 to 4th generation, and in case of penicillin, different types of. And the daptomycin, aminoglycoside, another class chemically. Aminoglycoside, generally the streptomycin, gentamicin, clindamycin, tobramycin, these are the aminoglycoside antibiotics. Another is the metronidazole, it is also antibacterial, both the aerobic and anaerobic bacteria. Another is the fluoroquinolones, it is the chemical class, but under this class so many antibiotics like ciprofloxacin, ofloxacin, sparfloxacin, lumifloxacin, levofloxacin, another is the vancomycin. So these are the example of antibiotics which are used for the antibacterial as well as the bacteriostatic and the bactericidal action. Next is the bacteriostatic. Those antibiotics that stop or inhibit the growth of the bacteria, means no multiplication or generation of bacteria. That means growth will be arrested and their multiplication or the reproduction will be arrested. That is called the, but do not kill the bacteria. Bacteria do not kill. That is called the bacteriostatic, and that is the basic difference between the bacteriostatic and bactericidal. In case of cidal, kill the microorganisms, but static, they did not kill, only arrest the growth of the microorganism or reduce the reproduction, slow their growth and the reproduction. Reproduction will be slow and growth will be slow. Inhibit the DNA replication, protein synthesis of bacteria, either by the DNA replication. I think this replication, that means DNA to DNA. The DNA is a very important molecules for the genetic material for survival of the microorganism, not only the microorganism, any living things. So that we inhibit the DNA replication, ultimately death, or protein. Protein is the very essential component for growth of the body. So that's why the protein synthesis will be inhibited by the bacteriostatic agent. Obviously, in case of bactericidal, there are the mechanism work with the immune system of the host, but here there is a difference. Bacteriostatic, sometimes they will affect the host cell immune system. Minimum drug concentration, here also same logic, the minimum inhibitory concentration is required. For example, tetracycline, one of the enzymes, macrolide group of enzymes, penicillin, sulfonamides, lisonides, chloramphenicol, clindamycin, and macrolides. But today question will come, the penicillin under the class of bactericidal, here also bacteriostatic. Actually penicillin group of penicillin, not only the one antibiotics, penicillin is one, but there are different class, penicillin G, penicillin V, penicillin and amoxicillin. So different types of penicillin. Normal penicillin, that is penicillin G, or benzyl penicillin, nowadays it is not used, but initially the benzyl penicillin come in the market, the penicillium notatum, the main source, but nowadays the commercial source is the Penicillium chrysogenium. The Alexander Fleming around 1929, he got the Nobel Prize of discovery of penicillin. Another is the clindamycin and the macrolides. Macrolides group of antibiotics, generally erythromycin, azithromycin, and clarithromycin, which are the macrolide group of antibiotics which are acts as a bacteriostatic agents. What are the mechanism of action? Number one, inhibition, target of the bacterial cell wall. Bacterial cell wall synthesis. It is the 5 types, 6 major type of mechanism of action. Number one, cell wall. Cell wall is the very important component to protect the microorganisms. If any means you can rupture the cell wall, immediately the cell will be destroyed. Ultimately the prokaryotes or eukaryotic, and the cell wall will be destroyed, then cell will be destroyed. That means they kill the microorganism. So it is the most important approach to inhibit the cell wall synthesis and to inhibit the kill of the microorganism. Number two, inhibition target bacterial protein biosynthesis, or protein in our body, or not only our body but microorganism, especially microorganism, they synthesize their protein in the body. They synthesize or biosynthesis of the protein. If you arrest or you stop or inhibit the protein synthesis. The protein is not synthesized. The protein is not synthesized. Ultimately, the death may be occur. And inhibition of the target bacterial nucleic acid. Obviously, nucleic acid is another component part, most important part by which you can stop the nucleic acid synthesis. Ultimately, death may occur. Target bacterial cell membrane. Another is the bacterial cell membrane, which is just below the cell wall. If the cell membrane will be destroyed, then all the organelles spread through the cell wall, and ultimately the disorganization of these organelles, and finally the death may occur. And inhibition of the metabolic pathway. These four are the very basic common pathway, but another pathway, inhibition of the metabolic pathway. Sometimes the microorganism, not only microorganism, others living organisms which are survive by their metabolic pathway, say for nucleic acid synthesis. By this mechanism, not directly inhibit the protein synthesis, cell wall synthesis, cell membrane synthesis, or nucleic acid synthesis. It's directly to stop a particular steps of metabolic pathway. Subglycolysis. Glycolysis stop, so glycolysis not occur, Krebs cycle will be not occur. So ultimately energy will be provided. So ultimately less energy to microorganism death. Inhibition of last term is the inhibition of ATP synthesis. ATP, it is the adenosine triphosphate, which is the high energy molecule, and that molecule is required for the living organisms. If you can arrest the synthesis of ATP, ultimately the organism will be killed. So bactericidal. First one is the inhibition and target of the bacterial. What is the bactericidal characteristic of the bactericidal? Inhibition, target of the bacterial cell wall biosynthesis. I already discussed. Target bacterial cell membrane function. Inhibition bacterial protein biosynthesis. Inhibition target bacterial nucleic acid synthesis. Bacteriostatic inhibition metabolic pathway. Basically, the bacteriostatic agent which are inhibit the metabolic pathway, inhibition of ATP synthesis and target nucleic acid synthesis and bacterial. Here, two different things: bacterial metabolic pathway and inhibition of ATP synthesis. In case of bactericidal and bacteriostatic are different, but the nucleic acid and protein synthesis is very common. In common both the case, bactericidal as well as the bacteriostatic. So in that case, how much amount of protein will be synthesized or inhibit the synthesis of protein or nucleic acid, that is depend upon this concentration of protein synthesis and the nucleic acid synthesis. You can classify it in it is a bactericidal or it is a bacteriostatic. Next, this is the bacteria, structure of the bacteria. The outer layer is the cell wall, then the cell membrane. And how will be what are the mechanism by which the drug or the antibacterial agent act upon this? First one is the be careful, be listen it. It is very helpful for your pharmacology as well as the microbiology and the biochemistry as well as the clinical pathology also. So be careful, listen it. First one is the and what types of antibiotic you can choose it and what types of disease or how to kill the microorganism, basic mechanism. I think it will be clear to all the mechanisms. Number one, cell wall. Cell wall is a very important component. If you destroy the cell wall, even if your cell wall or you are covering the skin, if your say 80% skin will be burned, immediately die because the infection will become. So here is the cell wall. It is a component of the bacteria that will be inhibited. That synthesis will be basically this cell wall is composed by the peptidoglycan in case of gram positive and in case of lipopolysaccharide in case of gram negative. But if any types of composition, if the cell wall is destroyed, the 99.9% sure that the organism will be killed or inhibited. And which type of antibiotics? Beta-lactam, like penicillin, cephalosporin, monobactam, carbamazepine, glycopeptides, vancomycin, and bacitracin. All are the very, very good bactericidal agents as well as the bacteriostatic agents. They basically inhibit the cell wall synthesis inhibitors. Next, the plasma membrane. Next step is the plasma membrane. This is the plasma membrane just below the cell wall. The polymyxin B, colistin, lipoprotein, and daptomycin, which are inhibit the cell membrane. Next is the cytoplasm. Cytoplasm contains so many compounds, and especially the protein synthesis inhibitor. Which is the protein synthesis? In case of protein synthesis, in case of prokaryotic, there is a ribosome is the factory house for the protein synthesis, and there is a consist of the two subunit. One is the 30S and another is the 50S, and the total is the 70S in case of prokaryotic. But in case of eukaryotic, quite different. It is 80S, 40S, and 60S. But here 70S, two subunit 30S and 50S. And actually, in case of protein synthesis, there is 30S and 50S both are required. So here some drugs which are bound with the 30S to stop the protein synthesis, and some drugs is bound with the 50S to inhibit the protein synthesis. 30S binding protein antibiotics are aminoglycoside like streptomycin. Just you can remember it or write down it. Aminoglycoside like streptomycin, gentamicin, clindamycin, tetracycline group of antibiotics, and 50S macrolides, azithromycin, clarithromycin, lincosamides, chloramphenicol, and oxazolidine. So these are the antibiotics which are directly bind with the 50S, and ultimately the protein synthesis will be inhibited. So any types of antibiotics which are either bind with the 30S or by the 50S, they inhibit the growth of the microorganisms. Next is the DNA synthesis. So DNA synthesis, that means DNA is the most important nucleic material which are growth, everything done by the DNA, and that DNA will be basically the DNA, bacterial DNA guidage or topoisomerase 2 to supercoiling the bacteria. In case of supercoiling, to prevent this there is the destroy of the microorganisms. So in this class, the fluoroquinolones derivatives, in this class ciprofloxacin, levofloxacin, moxifloxacin, sparfloxacin, and RNA synthesis rifampicin and rifampin. These are the class of drugs which are directly inhibit the nucleic acid synthesis and finally the death of the microorganisms. Either is a bacteriostatic or by the bactericidal. When bacteriostatic and viral, that will be discussed later. Another, in case of metabolic pathway, folic acid. Folic acid is the essential component of the bacteria. And this folic acid synthesis by the two step, one is the dihydrofolate reductase, another tetrahydrofolate reductase is enzyme is required for the synthesis of folic acid. So sulfonamides and sulfone generally inhibit this or bind this particular enzymes, dihydrofolate reductase or tetrahydrofolate reductase. Folinic acid to folic acid is converted to stop the folinic acid to folic acid. So ultimately folic acid cannot be synthesized, and if the folic acid not synthesized, because the folic acid the essential component of the bacteria, in that case the sulfonamides and sulfone that can inhibit the growth of the bacteria due to the folic acid synthesis inhibition. In this class, sulfonamides, sulfone, trimethoprim, and the mycolic acid which is present in the Mycobacterium tuberculosis and the Mycobacterium tuberculosis TB. So that are synthesized, especially the isoniazid, pyrizinamide, and streptomycin, which are inhibit the synthesis of mycolic acid, and mycolic acid is essential for the growth of the Mycobacterium tuberculosis. So inhibit the mycolic acid, ultimately the death of the tuberculous bacilli, and finally to recover the tuberculosis. Okay, these are the based on the mechanism action. We can discuss elaborately. If any question, please ask me up to this today. Up to this next day, I discuss in the later part of this. Okay, thank you very much for listening to the lecture. Okay, thank you. Another question. Here is the 30S and the 50S. What the full form the S? Anybody can tell? What the full form of the S?